we are recording this chat just to try and keep people in the loop. There are a bunch of folks who are interested but weren't able to attend today, and so we're trying to build some community and promote, communication and harmonization across different studies that are happening right now in the space of trying to understand pulse oximetry disparities and improve pulse oximeter performance and equity. So this recording we will post to the community forum on the Open Oximetry website, just so you know, in terms of what you're sharing as relates to any kind of confidential data or otherwise. So with that, the recording has started. I'm going to give just a very quick overview and then hand off to my colleague, Dr. Carolyn Hendrickson, who's going to lead most of the discussion today. For those of you who I haven't met, I'm Mike Lipnick. I'm an anesthesiologist, intensivist based at UCSF at San Francisco General Hospital, part of the hypoxia lab, and helping to lead the Open Oximetry project, which is a combination of laboratory-based tests and clinical studies to try and understand oximeter performance under a variety of conditions, including this issue of skin color, as well as low perfusion and other things. As part of the Open Oximetry project, for those of you who haven't already participated, we're working very closely with some regulatory bodies like ISO and FDA to try and figure out how to raise the bar for safety with pulse oximetry. And as part of that, we've been having a lot of meetings, some of which have been larger meetings, some of which have been smaller meetings like this, to try and figure out how to harmonize the many different efforts that are happening in parallel right now to see if we can accelerate finding answers, because the answers to the questions we all want are taking a lot of time, and there's a lot of urgency to try and figure this out. And so this particular group is focused on teams that are pursuing already or thinking about doing clinical trials, prospective data collection in patients who have blood gases and pulse oximeters on, to try and figure out how those data can help us answer questions that we can't get out of either laboratory data or out of retrospective studies. Part of the purpose for today's meeting was to, in follow-up to a prior meeting with this group, provide very brief updates to what people are up to at their study sites, to try and figure out data quality issues and to share some challenges that our team is having, and also see what challenges others may be having in an effort to try and understand how we can improve on that, but also to try and figure out if there may be opportunity, as discussed in prior calls, to harmonize studies in such a way that if we collect the same quality data in the same way, that even after our individual groups may publish those data individually, we may be able to pool the results to answer questions more definitively. That certainly has been the experience that we're having in terms of wishing we had more data, as we all always do. But so we wanted to think creatively with everybody on this call about ways we might be able to do that, including whether or not there may be additional study sites that we could help support with kind of the resources that we have, which may or may not be enough to do that, but to just try and brainstorm a little bit about whether or not we might be able to accelerate finding answers in this space. So with that, I'm going to hand it over to Dr. Hendrickson to lead the discussion. I apologize, I'm going to kind of be in and out on some unexpected clinical duty. But again, thanks everybody for taking the time to join, and look forward to the discussion. Carolyn, over to you.
Yeah, thanks, Mike. Welcome, everybody. I'm going to share my screen, and this opener slide is really just in case anybody wants to use the QR code to get to the OpenOx study site. So all right, so that's how is that for you guys? Can you all see my slide? Yeah, thumbs up. Okay. So one thing I'd like you all to do as I start walking through some of our slides is that I wanted to invite people to share a snapshot of what's going on at your site right now, or what might be in the works getting started. And then we can kind of identify some of the barriers to collecting data that's aligned with the OpenOx protocol, and barriers to sharing data between sites, so that we can answer these really important questions in a timely manner. If time allows, maybe we can table some of the group discussion for the end after we get a survey of all of the different sites, just to get a kind of bird's eye view of what's happening in each place. So I am going to just remind you that there's a lot of work going on in pulse oximeters. The question that we're trying to understand now is how do these devices interact with different skin pigments and generate potentially different kinds of errors for different patients. And to date, we have a lot of great data from some of the people on this call showing that there's an association between race and differential pulse oximetry errors. But really what we want to understand now is what is the mechanism there? And, you know, I want to acknowledge that it's probably a lot more complicated than that first box diagram I showed. But the minimum kind of data set that we need to answer the questions really does collect objective and subjective skin pigment data. So we're hoping to identify work that's going on that has a minimum data set of these subjective or objective skin pigmentation captures and like simultaneous observation of paired SpO2 and SaO2 samples. The retrospective work is really powerful. It's been a wonderful groundwork for studies going forward, but we think that we're all now looking at prospective studies. So I want to call out that you know, we want measured functional SaO2. We want the ABGs to be paired with an observed SpO2. There's some data from the monitors also for ICU patients that we'd like to collect, including perfusion index that the device is reporting, and the stability of the reading as well. And then we're collecting a variety of skin pigment assessments at the SFGH site. I'll say that the von Luschen and the Fitzpatrick scales are woefully inadequate. And so the you know, future data collection is really going to make sure we prioritize collecting the Monk skin tone scale. And I see Dr. Monk is on this call as well. And really, we want to make sure that we have a good distribution of observations across all of the skin pigments that are captured by this scale, and particularly enough observations in the darkest 2 to 3 categories to make sure that we understand how the device may interact differently with different skin pigments. I'll also say that the spectrophotometer data that we're collecting for us, we're using a Konica Minolta, which is in the top left, as well as Delphin. We've been doing some comparison between these 2 data capture methods, and we're probably leaning more towards aligning with some ISO discussions about using the spectrophotometer to capture data. So we're also collecting skin color at multiple sites. You know, it feels intuitive that it's the skin pigment at the site the oximeter is working at. That is a really important question to answer. And I don't know about you guys, in our ICU, we have most of the probes on the finger, but in patients who are having a really hard time getting signal, like there's a lot of shopping around for a site like ear, nose, changing from a reusable to a disposable device. All of these things may impact our findings. So we're trying to collect skin pigment data from as many sites as possible, just to have a rich data set. This may not be feasible for everybody's site. The other thing that's challenging for us, and I'm sure probably for everybody, but we'd love any tricks that you've developed, is that finding desaturation events in the ICU of less than 90 years is kind of rare because people get rescued. So, in order to observe those events, we really need people at bedside collecting these data. And then additionally, FDA is interested in collecting data from multiple research devices, not just the standard of care device, but certainly collaborating with other research groups that use a different oximeter set for their standard of care would be really valuable and of interest. We happen to have Massimo's at both of our clinical sites. I think I already touched on this. So, we've got some fairly complete data sets, but occasionally it's really hard to get this device on someone if they've got a C-collar on, for example. We might not have data for the earlobe site. So there's just some pragmatic considerations when you're using a giant spectrophotometer like this on an ICU patient with gear on. Okay, so these are the questions I'm going to pose to the group. And if you could drop your site's name in the chat, just so we can have a little roll call. I understand that not everyone is enrolling, and some people are here just because they want to learn and they'd like to be involved, and that's great. But I want to make sure that Fifi and I and Mike are attending to the chat and get a chance for everybody to share what's going on with you. So we're going to kind of ask you to tell us about enrollment. Have you started, or when are you planning to start? Do you think that you're going to get sort of at least a steady stream of 2 subjects with 4 ABG samples a week? How feasible is it to collect subjective skin pigment data and objective skin pigment data with a spectrophotometer? Are you in need of assistance purchasing a device like a Konica Minolta? We'll scrap the Delphin skin color catch for now. And then maybe if you could just estimate, or you have these numbers handy, what percentage of your potential subjects might be in the darkest skin pigment categories? I put the Monk skin tone scale above, but maybe the darkest 2 or 3 groups. And then if you have any funding horizons or anticipated grant mechanisms that are coming through, we'd love to hear about that. So I'll just report out for us. We're enrolling patients at the UCSF affiliated county hospital. It's a pretty small place, which makes it nice because all the ICUs are on 1 floor. And so in the first 10 months of enrollment, we've enrolled 226 fully consented subjects. We're hoping to get up to about 300 by August. We have currently 550 blood gas samples with paired SpO2 observations. Only 71 of these are at saturations less than 90%, and only about 5% of our subjects are in the darkest 2 categories of the Monk skin tone scale, and it goes up to about 8% if you include the top 3. So let me just see. I'm going to open the chat here. All right. So kind of in order of response, maybe Dr. Jalilian at UCLA, do you want to start with any updates from your site?
Yeah, hi, do you see me? Hello?
I have a lot of screens open, so I don't know if I see you.
Oh, okay, all right.
Yes, we do.
Okay. Oh yeah, I see you.
Again, at UCLA. It's funny, I saw the UCSF clinical data collection protocol after I developed my own protocol, and it was almost identical to what you guys developed. I modified mine a little bit just to make it almost identical, like completely identical actually to your study, Caroline. So I would say I'm collecting all the data that you're collecting at UCSF. I've enrolled 32 patients. I haven't limited myself just to the ICU. I collect in our cardiothoracic ICU, where I take care of, post-transplant lung, heart patients. I would say I've gotten some good saturations in the 80s. I also go into the OR and have enrolled pediatric congenital cardiac patients who are in the cath lab for diagnostic studies. So I have a large number, maybe like 12 of my patients right now have sets in the 70s or 60s, and those are the babies. But I've been having the same issues as you. These patients who desaturate get rescued immediately. So the ICU data is very challenging to get, which is why I shifted more into the OR to get data on the congenital cardiac kids. We have a high volume here at UCLA. I'm getting this data to put into an NIH grant. Right now it's just me collecting, but I'm going to enroll some medical students or get some medical students and residents to help me. So I anticipate scaling this. I get data maybe on 7 patients, anywhere from like 5 to 7 patients a week. Our OR volumes are high, and then the ICU, just whenever I'm non-clinical, I go in and I get data. But I think once I scale myself, I can expand it more. Just right now, I'm trying to figure out like how it works to get data, and then I'll trainees.
And you're getting both the subjective and the objective skin pigment data, and if so, what device are you using to capture the?
I'm using the colorimeter. I'm not using the spectrophotometer, the Konica Minolta. I'm just using a colorimeter, the Fitzpatrick skin tone, Monk skin tone scale. And then the Von Lucian, honestly, like it's very hard for me to, like distinguish. So I initially was collecting that data, but I really don't think I'm doing it well. And I didn't print it out the way you guys like laminated it and printed it. So I don't have that data, but I have Fitzpatrick skin scale, the colorimeter, and I enroll patients across all ICUs. We have 8 ICUs at UCLA. It's the colorimeter you guys use. I saw what you've posted online. Like I said,
the Delphin
I'm getting is pretty much equal to this data that you're getting. And we have 8 ICUs and all the ICU directors know about this study. So I think once I scale myself, I can really expand it more.
Great.
It's just, I'm still figuring.
That's exciting.
Yeah.
Thank you. That's really exciting. Yeah, we found that the Von Lucian, like nobody's skin looks like that. So it is pretty hard to like take your best guess at which bucket they're in. Okay, thanks for that update. So let's keep moving through the roll call. Next, I have Brady Scott from Rush.
Yeah, so we are currently IRB approved for our study. We have not yet enrolled anyone. Currently, we are using the Monk scale. I have been seeking, I think I have been approved for at least some bit of funding for a colorimeter or the Konica Minolta device. As I've heard some others had a little trouble with the colorimeter, the Delphin or whatever it's called. We have not yet enrolled anybody, but we are, I've assembled some of my graduate students and I have a med student that's interested involved. So we're trying to be able to reach more, get out there with more patients. We are currently, what we were going to use in terms of devices were the, oh shoot, I had it pulled up in front of me. I just want to make sure I say this right. It's the Massimo Rad-G pulse oximeters, and then the Medtronic Nellcore portable PM10N. And then in our ICU, our patients are on Philips monitors. So then it would have 3 different monitors or 3 different brands of monitors. And then currently using the Monk scale the way that our proposal is currently approved. I've just got to, I have to be honest with you. I didn't have the dollars laying around for $12,000 for a Konica Minolta device. So I had to go after a little funding for that. So I'm excited to see, I'm willing to, I'm excited to see how we can contribute or even change our study to make it more usable into the larger data collection. So thanks for having me here again.
Great. Yeah, thank you for the update, and understood it's hard to find $12,000 laying around for equipment. Okay, next on our roll call is Joseph Reinhardt from UC Irvine. You may not be able to unmute, so I'll come back to UC Irvine. How about Christopher Allman from Stanford?
Yeah, actually, thanks very much. Rohan's going to give a little bit of an update here with the guide questions. Thanks.
Great. Thanks, Rohan.
Sure. So at Lucille Packard, we are enrolling, and I want to first thank the UCSF team, because our protocol is very similar. To what, so we use both the Konica and the Delphin right now. We are using the Von Lucian, the Fitzpatrick, as well as the Monk scale. We've enrolled about 103 subjects as of Friday for a total of 368 samples. And I would say of that, about 40% are less than 90% saturation. We are recruiting in the CVICU and the cath lab primarily. But yeah, I think, and similarly, finding darker skin tones has been a challenge. We've in the darkest of I would say in the darkest of the 3 monk scales, we don't have anyone right now, and in that 29 plus on the von Lucian, we probably have 1 subject. So that continues to be a challenge, and we are using the Massimo Rad7. That's the only pulse ox we use. I'm trying to think if I miss any of your questions, Carolyn.
I think you hit most of them. And do you have any plans to change your recruitment strategy? And for us, we sort of like recruit everyone, we approach everyone. So we haven't really had too many levers to pull to access, the patient population that may be most affected by these errors, the folks with the darkest skin pigmentation, but I don't know if you have any differences like community engagement or anything like that.
Yeah, that's a great question. Chris, do you want to add something there?
Yeah, no, yeah, it's interesting when we looked initially at our demographics, we thought we would be able to cover the spectrum. But I think what we're really learning is that for those kind of category 4 patients in von Lucian, so with a scale of 29 and above, that most of the patients who self-identify as Black don't actually end up in that end of the scale. So we have had discussions about going outside the heart center, but we think the problem is probably more institution-wide than just the heart center. So we have had some thoughts about perhaps including another site, but obviously that would come at considerable cost to do. We've thought about Oakland, but we're still not sure that would necessarily provide more category 4 patients. We've also had some transient conversations about thinking of either other cities or overseas sites that might be doing similar work where we could enroll. So we've mostly just been having some brainstorming on this. We have our next meeting with the FDA coming up soon, and I think that will be probably the most important topic for discussion.
Yeah, they were very interested in that from us as well. Okay, well, thank you for the update from Stanford. I just want to circle back and see if Joseph Reinhardt from UC Irvine is available to give us an update. All right, let's move forward with, looks like Garrett Burnett from Mount Sinai.
Hello.
Hi.
So I'm an anesthesiologist at Mount Sinai in New York. Most of what I've done thus far has been retrospective in the anesthesia world. I did put in for an Anesthesia Patient Safety Foundation grant to look into this to try and help fund buying the Konica Minolta $12,000 or how much ever it costs. And with the idea of initially just looking in the congenital pediatric population, but with the idea of hopefully trying to be expanding this to ICUs similar to what other groups have been doing. My plan is to more or less try and conform to this kind of standard protocol so that we can put our data together later. But at this point, I'm waiting to hear back from the grant, and I don't know. I think I'm quite a bit behind most people in this group, but hopefully, because where we're at in New York City, we have a pretty diverse population in terms of skin tones.
Yeah, that's you're not so far behind. We hope that the OpenOx protocol is exportable. So maybe at the end of the call, it would be useful to understand like if that is in fact true for sites who have tried to do it. It sounds like a couple people have. And really it's sort of your site specific IRB approval and staffing and equipment that are the major barriers to enrollment. And I don't want to minimize again that $12,000 expense for for the for the colorimeter.
Yeah,
for the
I'm hoping to hear back
spectrophotometer.
from the grant soon so that we can kind of start moving forward. But thank you.
Okay, great. All right. Thanks for that update. Let's see, who do I have next in the chat? It looks like Dr. Fozzie from Johns Hopkins responded.
Hey there, everyone. So sorry, first of all, because I'm in the lab right now, so it might be a little loud in here, so I hope everyone can hear me. So Ash Fozzie from Johns Hopkins, I'm a pulmonologist here. This isn't my primary area of research. My research is in COPD, so this is kind of a side project for me. As some of you may know, we did retrospective work in COVID-19 before in this topic. I started a prospective study in June of 2022 that was somewhat similar to the one that this group came up with in terms of looking at skin tone objectively. We bought the Konica Minolta colorimeter, so not the spectrophotometer, but the CR400, I think it is, which is $8,000 instead of $12,000. And we enrolled 12 subjects between June and October, I think, of 2022. And then we had to stop because we didn't have any funding. The staff that I was borrowing from our ICU group had to do other projects, and then one of them left. So our study had been paused since then. I also submitted a project as part of our U01 CERC grant, and while the grant was renewed, unfortunately, they did not fund any new projects. So our relatively paltry ask for money was not approved, so I don't actually have any funding for this. There is potentially a chance to restart because the ICU groups staff, it doesn't have as much to do as they did before, but I'm a little hesitant, honestly, to restart because our recruitment was actually pretty difficult to do in terms of finding people who actually agree, finding their someone to consent for the patients who, were unconscious or couldn't consent for themselves. All of that was pretty difficult. And one difference in our protocol compared to the one coming out of UCSF was that we are actually depending on clinical data. We do have a system that records all of the data that comes through the monitor so that we can find out what the pulse ox was after the fact at the same exact time that the ABG was drawn. So, we weren't drawing ABGs in addition to the clinical ones. We weren't having anyone on site to check pulse oximetry at the time or anything like that. So that's also made me a little bit hesitant to restart because I don't think we can go all the way and have and put as much into it as the UCSF group is, so that our data would really be similar to that. In terms of subjective measures, I mean, we started this before before the protocol came out, and honestly, before I knew about the Monk scale. So we were only using the Fitzpatrick in addition to the objective measure. But obviously, if we restart, we would use other subjective measures as well.
Okay, thanks, Ash. Yeah, I hear all of those challenges, and not having a funded CRC team or any funding is certainly a barrier to getting research done. So thank you for the update and for calling attention to those important considerations. Next, I have Benson Chen, and I think Farzad Moazed is also here from Alameda Health System.
Hi.
Nice.
Nice to see you. Sorry, I'm on service, so it's a little chaotic. We've chatted, obviously, Carolyn. I'm Farzad. I'm at Highland Hospital in Oakland. Works with Carolyn a lot back. Additional investigation site, basically. that is very much in line with this type of study. And yeah, I don't know how to answer these questions, but I think, you know, we're hoping to be an enrollment site. We have a lot of patients that would be potentially eligible. I don't have actual figures. I'm sort of on service this week, so kind of looking at that, but we have a lot of patients with A lines and certainly a lot of patients with Monk scale scores in the last 3. I'd say the maybe not the last one, but the 2 before that, we certainly have a lot of patients in our ICU with those tones. I don't know if there's anything you want to add, Benson.
No, just appreciate the opportunity to be part of the discussion, and eager to participate if we can find the resources and funding to do it.
Yeah, thank you. We are eager to keep those explorations going. Fifi, thanks for calling this to my attention. Sorry, Dan Franklin, I skipped you from Toronto. Do you want to give us an update on your site?
Sure, no worries. Hello, everybody. My name is Daniel Franklin. I'm an assistant professor here at University of Toronto. I personally am an engineer. So we have 2 protocols that we're hoping to act on, one of which is in review. It's an internal one at U of T for recruiting healthy volunteers. However, the big sticking point is that we think we may have to go with the silver standard as opposed to the blood gas. So for that effort, also, we've put an application to the OpenOx call for devices, because we make and manufacture devices that we think can help alleviate some of these disparities. The other effort that we have going on is exploring separate protocols at Toronto General and SickKids and trying to find clinicians to buy into that process. And so that's where we're currently at. Regarding the devices that we use, we have a massive radical for the claimed silver standard. They claim that theirs is relatively independent of skin tone, but we also have a Finipres Nova for a whole range of hemodynamic measurements that we're looking to explore simultaneously for the healthy population. And I think the other thing is regarding the skin tone categories. We're just hoping that with Toronto's diverse population that we can achieve adequate binning.
Yeah, thanks for that update. And forgive this question. You mentioned that you don't have arterial blood gases. Did I hear that correctly?
For the internal U of T one that my lab itself is conducting, because my engineering background, we probably have to partner with a clinician in order to perform those.
Gotcha. Understood. Thank you. Do you have a clinician investigator at Toronto who has the experience for clinical research that would be able to partner with you?
Yeah, my closest collaborator is Heather Ross at the Peter Monk Cardiac Center. We're just trying to find a place to fit all the pieces together, basically. So, where would be the best clinical population to conduct the study? And so I was curious about hearing the different populations that were explored. We're also looking for the healthy volunteers, somebody to conduct the step down hypoxia as well, because that's not a common practice within the cardiac unit here.
Okay. Gotcha. All right. And then I'll call UC Irvine one more time. Is anyone from Irvine available? Joseph Reinhardt or other team members to give an update? Okay, so maybe not available to come off of mute right now. Did I miss any potential currently enrolling or planned to enroll study sites that anybody wants to chime in about? Okay, please feel free to interrupt if I missed anyone. So, we've got a pretty wide spectrum of work going on, with some folks preparing for potential enrollment, other people needing to identify additional resources before they can get running, and a couple of sites that have berries around a specific funding source, like for Ikonica Minolta, but otherwise would be able to kind of do the lift with the resources they've got currently. And it sounds like Stanford and UCLA are both farther along and have consented and enrolled a number of subjects and are looking to either more effectively recruit or expand to a second site, or kind of for Dr. Jalilian, get some more people involved in the lift. Folks have mentioned some really pragmatic barriers to executing the research. I see some chat about device cost. And I also wanted to see if there's anything in specific that people want to bring up that are solvable problems, maybe that we might be able to think about together, either with implementing a protocol or identifying resources. I think it sounds like a lot of people are funded through FDA and CIRSE, or just kind of doing this out of a labor of love. Brady, I see your hand is up.
So I had a question about for those of you that are using these subjective scales currently, like the Monk scale, what kind of, are you doing any kind of special lighting around that? Are you using any kind of, or are you just using room lighting? That's kind of a conundrum that we've written into our protocol. We actually bought a special light for it to use that we thought would be a little better here, but we're not sure if that's what everybody's doing.
To shine on the skin, or and so that you can compare the standardized printout? To
shine on the skin when you're trying to make a subjective analysis of which one of those Monk scale colors, which would be the most appropriate to say the patient has or is. And we're just making sure that we're just following whatever makes sense. Is everybody just using room lighting for that? We had actually one of my students here who's on the call is a dentist, actually, and is very sensitive to matching teeth during when he uses like crowns and things, and they use a certain kind of light source for that. And I just didn't know if that had been a consideration or if it's something that we should be considering.
Oh, that's such a good question. I'll tell you that we're pretty pragmatic and use ambient lighting, but it sounds like this is something that we should be considering. Sorry, I can't read your full name. Yeah, are you the Maciek? Are you the dentist?
Yes, I'm the dentist. So the theory behind it is that the light that the tone of the skin is reflective of what light source you're using. And so in dentistry, in order for us to ensure that we're properly matching the tooth color, which can be very important, we are required to have 100 lighting that's rated at 100 CRI. So it has a chromatic reproduction ratio that's 1:1. You can buy small lights that can match that reproduction ratio, because when you have a light source that isn't matching up to at least 80% of the CRI, what'll happen is that you'll have a incorrect comparison. So even though the shade guide and like in this case the tissue may be visibly the same color when you're comparing it, unless it's a high rated light source, or like sunlight, which is a cheaper alternative if you have good access to daylight, it'll under different light sources it'll look completely different, and you may think you have a match when in reality they didn't match at all.
Understood.
So like Dr. Monk can chime in here.
Yeah, Dr. Monk can expertly comment on this. Yeah. We do have daylight in our ICU bays, but I'd be really curious to hear what you have to say, Ellis.
Yeah, so my experience with this is that I think a lot of people just kind of use whatever light they have. But I'm very sensitive to the point that was raised here about lighting. And I think one of the things that ISO is discussing right now is requiring the use of a full spectrum lamp as part of the protocol with subjective color matching with the Monk scale. So I do think that going forward, that's going to be one of the things that's kind of wrapped into the protocol, is that there's some kind of lamp, full spectrum lamp light source that's used so that everyone is on the same page. That being said, I haven't noticed any kind of like wildly different annotations with the lighting being different. In fact, some of the testing that we've done at Google, we've actually altered the light of photos and even videos of people who are being annotated, and we still have high rates of consensus with our annotators, even in those cases where the images are poorly lit. So I don't think there's room to be, that concerned, but of course, with the scientific method and the protocols, you want everything to be as equal as possible. So that is something that's being discussed.
Okay,
Thank you,
Dr.
Thank you
Monk.
so much.
Yeah.
How much did your little device cost?
We currently again, we haven't enrolled anything, so we may have to change, but we bought a Loom Cube. That was what Mache said we should buy, and I think it was what Mache paid, maybe $50 or $75, or it's a little little thing. The question is I just remembered the last conversation we had. I heard people say we're using ambient light, and I just wasn't sure if maybe you had investigated that, maybe what we're doing is a bad idea, if it's a good idea. And I just want to, again, make this somewhat, useful at the end of the day for everybody to be able to use our data. So it was inexpensive.
Yeah, that's a great point, especially if it's inexpensive. It seems like something that we could improve our protocol with.
Dr. Monk, do you have those lights that you were referring to? Do you have a list of those lights, or
I can figure out what the list is. I do think that the price point that you mentioned is something that was mentioned to me when they were discussing this as well. They said it was around $70, so I'll get that list for you.
Oh, thanks. I appreciate it.
Yep.
Lale, were you unmuting? I wasn't sure if you had a question or comment.
Yeah. Just my own experience going into the OR and the ICU. I kind of took a very pragmatic approach as well, just whatever the room light is. When you're in the OR getting the data, you don't want to really interrupt the flow of the anesthesia and surgical teams. Sometimes I have to really quickly get in and get out. So I hadn't thought about getting a lamp. I felt generally like the ambient lighting has been pretty sufficient. And then, there's the broader question of how granular and like how granular do you want to be on these subjective scales? I don't know. So that's my 2 cents. Thanks.
Yeah, I mean, I think one of the strengths of Dr. Monk's skin tone scale is that with 10 categories, there's enough distinction, but it's not overly burdensome with, like 30 plus pigments that don't actually look like anybody's skin. So I think, I don't think it's overly granular, but I also really appreciate the comment about, subjective assessment being pretty reproducible even in lower lighting. So understood about the OR. Clinical research is messy, but maybe like if you had access to the forehead or something during a time of study, or even post-operatively, that could be like in the PACU, that might be of interest. I see your hand is up again, Dr. Monk.
Yeah, just really quickly to that point. So yeah, in constructing the scale, we wanted to make sure we didn't go beyond 10, mainly because most of the research shows that once you go beyond 10 or 12 points, things become very unreliable, and cognitively it becomes too much for people to make these fine-tuned distinctions. One thing to think about with the scale that I like to try to make clear for people is that when we're testing for consensus at Google, and Meta, Facebook did the same thing, the gold standard in data annotation and artificial intelligence and machine learning is plus or minus 1 point on the scale. So if people are not precisely on the right swatch, so to speak, perfectly fine. The gold standard in those worlds is plus or minus 1. And the binning that's being discussed at the level of the ISO also keeps that in mind, that, people are not going to be absolutely laser focused, precise. That's not really what the subjective measure is supposed to be about.
Thank you for that comment. I don't see anybody else unmuted or raising hands. I'd just like to invite any other comments or questions or points for discussion that folks want to bring up.
I was wondering if there's any plan to make this into a true multi-site project and apply for a multi-site grant, potentially through the NIH or somewhere else. You know, I just think of the multi-site COPD studies that we have, and you know, there's always the pedal network and stuff like that. That, you know, I feel like if these funding agencies see that we're doing this without funding, they'll say, well, you don't need funding for it anyway, you're doing it already. I know that you guys have funding at UCSF through CIRSE, but I wonder if there is any plan to make this a truly multi-center project.
Yeah, I mean, I think that would be ideal, right? To have multiple sites, multiple devices, multiple populations really well represented in the data. We are trolling funding mechanisms and haven't seen anything that would cover, you know, making a network. I think the hope might be that with initial data analysis and publication, we might, you know, be able to raise enough questions about mechanisms and performance improvement that we could make a case, because the fund, I mean, it's mostly salary, right? That people need, like for clinical research coordinators, a little bit of PI time, and then there's an initial upfront investment on equipment, but then it gets, you know, a lot more feasible going forward. So I would really invite any ideas for avenues for advocacy, but you know, we share that as a sort of an ideal scenario. It would be great to have more formal networking going on.
Yeah, I wonder if a specific discussion with the NIH about this would make sense in terms of they can make a mechanism that is specific for this, or make it into a contract or something for a multi-site observational study, because you know, it's not really going to fit into any of their RFAs or anything straight away.
Yeah, I will have to educate myself about how we could formally advocate, but part of the reason we wanted to convene is just to understand some of the barriers, which are, you know, what we said, it's funding and time, and people need to be compensated. And even though lots of folks are kind of doing a big lift because this is important, that's clearly not like the most efficient or sustainable path forward. I just want to call out in the chat that Rohan was asking about Delphin and you know, are we still using it for data collection? And we are, but some of the analyses that the ISO has been using to compare the Delphin and the Konica Minolta suggest that there might be some problems with calibration and consistency. So even though that device is like a smaller profile and less expensive, making it attractive for those reasons, it may not be generating the same quality data.
Thank you. Thank you.
Okay, so I think Ash is asking the real important big picture question, which is like, how can we be most effective as a group? And I think funding and advocacy are something we could turn our attention to once we got a little bit more of the sort of logistics under our belt. We, as you mentioned, we are funded by FDA. And we're looking at opening 1 to 2 more clinical sites within the UCSF system. And I think once those hurdles are cleared, we should be more forward thinking, as you're suggesting. But in the meantime, maybe we can help limp some of these like really noble efforts along by identifying funds for, say, a Konica Minolta for sites that are otherwise kind of ready to go and contribute to a data set that would functionally be multi-center. Okay, we have 7 minutes till the top of the hour. I just want to make sure that I've invited everyone to join the conversation and ask any questions or raise concerns that they might have.
I'll ask one, Dr. Hendrickson. So if I recall correctly, on your protocol, your consent kind of goes something like this: try to get consent, then go for a family member or legal some sort of legal representative, and if not, still go ahead and do the study, and then follow up on the patient until maybe either discharge or another outcome. Is that correct?
Mm-hmm.
So that's exactly what I have to do. That's my IRB led me down exactly the same path. I don't know if you're willing to share. I don't know if not, that's okay. How you document following up. If you do that every day, do you keep that on what kind of document you use for that? I got to create something for that I have not actually been approved on yet. And I don't know. I'm willing to ask to not reinvent the wheel. It sounds like I'm doing exactly the same consenting maneuver or mechanism you are. And
yeah, and we'd be happy to share. It sounds like you have seen the consents, and just for the group, they're available on the OpenOx site. We'd be happy to show you. We could maybe pull like the PHI from this email that we send every day. The research coordinators reply all and say like, these are the new enrolls, these are samples we got, these are the consent changes. And then they also make notes in REDCap. So you can do kind of like a note to file, like, I talked to the patient's daughter, she's too overwhelmed and asked me to come back in 3 days. Next check-in due on, June 1st. So we'd be happy to share what we're doing by sending you some screenshots of stuff that redacts the PHI.
That would be great. And if you're not willing to share it, or somebody says, hey, don't do that, that's okay. I just, this is the first time I've consented in this way, where I can go ahead and do the study, then I have to follow up repeatedly. And I just kind of, and I've asked for some other researchers around me if they've done that before. They haven't. And so it seems rather new, at least in my circle. And I just was going to see if there was a model that we could see. Again, it's up to you.
And if people have the same question, we'd be really happy to share. I mean, the balance we try to strike is to not be annoying, because families and patients are burdened with a lot of stuff going on with their care, but to give them the opportunity to tell us when it's okay to talk about it. And so we try to make ourselves available when they're ready, and also give them like an out if they're like, I cannot talk to another person today. And our IRB, I think, wants us to strike that balance. They don't want you like in there 5 times a day, like, can we talk? Can we talk? So we try to just kind of read the room a little bit, and then document any contact or any time that the nurse is like, you know what, she's really overwhelmed right now, can you come back? We'll document that as like a effort that was turned down by the clinical team. But yeah, we can share that. No problem at all sharing that kind of information.
Thank you. My team and we appreciate you for that.
Yeah, I mean, well, we don't want to have to reinvent the wheel, and we also want, if you got iterations that are better than what we're exporting, then please tell us, because we want to, like your suggestion about the light is really important. So we truly do want to get better from everybody else's experience too. Okay, so we are okay. It looks like Ash is gonna see the documents online. And Ash, if you have any questions about kind of like the logistics of implementing that, then please let us know. And then Brady, we can send your team an email, and you can take a look and tell me if I've got it right, and if you need more information than what I send you. All right, so I want to I want to wrap up, I think, unless there's any like last burning questions or things that you all want to connect with via email. Okay. Thank you all for your time and all your efforts to date. We really appreciate the work that's been done in this space and that everyone is so interested in answering these important questions. I have a to-do list to follow up with Rush, and please stay tuned for future meetings, and do reach out if there's anything that we can share with you that would be helpful in getting your site up and running.